All right. I welcome everyone for this eighth lecture, and I've been given a tall task of introducing the today's uh, who happens to be my teacher also now because I have done a
Oh
course under her,
God, please don't tell like that.
and she's Dr. Gargi Roy Goswami, Director of Coronas Private Limited, Founder Director of Indian Academy of Clinical and Dental Genetics, Founder and Chairman of Saliva Symposium India 2019, Coordinator for International Board of Saliva Diagnostic Science Denmark, Adjunct Faculty Research Development, Savita Dental College and Hospital Chennai. Now, this might appear a little brief because it's very difficult for me to concise the kind of work she has been doing. She has been doing a very passionate work in the field of saliva diagnostics and its applications. And that's what is the topic today. It's an honor to have you, ma'am, on this platform. And uh, apart from the scientific work she's doing, she runs a very innovative, informative thing, which is where she has interviews of people who are very renowned in their field, and she runs on a YouTube channel in that. And it's a privilege to have you, ma'am.
Thank
And
you so much. Thank you.
without wasting time, we will put her on the thing, and so that she can really share her experience in saliva diagnostic and its applications. Welcome, ma'am. And
Thank you.
before that, yes, yes,
Yeah,
yes.
yeah,
I need to really highlight people behind this thing.
sure.
The team is the same. Myself, Dr. Priyanka, Dr. Deepika, Dr. Pooja. We all work together, and we have been managing somehow to get very excellent people who are doing very well in their field. And one of them in the entire constellation is one of the stars which we have got today.
Oh
Welcome, Dr.
God,
Gargi.
I'm a very small person. No need to tell so many things about me. Okay, so I'll just share my screen
Yes, yes.
now.
I'll stop sharing first.
Um, it says host disable
Yes, yes,
participation.
yes, yes. Now you can.
Is it visible on my screen?
Yes, yes,
Oh,
yes.
fine. Okay. So, good evening, everyone. And I, Dr. Gargi Roy Goswami, with a mission to create, collaborate, and contribute to science. Today, I'm here to present in front of you. And before everything, I would like to give my prayers and gratitude and love to my dad, who is my hero, Dr. Kamalesh Chandra Roy. He was the first dentist of district Murshidabad and town Berhampur, West Bengal, India. That is my hometown. Okay, and I'm really thankful to the very innovative team of innovative pedagogical learning and for giving me this opportunity. And also, I extend my thanks to Indo-Pacific Academy of Forensic Odontology and Association Forensic Odontology for Human Rights for extending their support and making this this platform a wonderful knowledge sharing platform. I really appreciate the efforts that all of you are taking. So with that, let me just go into and give you the GPS of today's saliva diagnostics and its applications. So this is the lecture GPS. I'll start with a brief introduction to the body's mirror. That is nothing but saliva. And then I'll give you a little bit of drone view of saliva application, meaning to say that in what areas, saliva has its role. And I will also try to tell you in very brief that what is this world of omics, the things that we have been recently hearing much. And also, I will focus on certain prominent technologies of how to analyze the saliva as a biological sample. And then I am not expert in the forensic field, but I will definitely, I have tried my best to just add on my little knowledge and some information that I have with me regarding saliva being used as a forensic tool. And also, I will try to highlight the potential areas of research with saliva and dentistry, because I think if dentists, always I say that if dentists start seeing saliva as a sample, wonders will start happening. And then I'll tell you a very interesting thing where you can move, you can think of taking up your academic innovations or your academic work towards entrepreneurship. And then that's the red signal where I'll just complete it with little bit endeavors that I do. All right, so let's get introduced to the body's mirror. I hope all my slides are visible to everybody and I'm audible to everyone.
Yes, yes, yes.
Okay, thank you, Dr. Amit. Okay, so we all know what is saliva. It's basically a slightly alkaline fluid that is secreted by our salivary glands and also mucous membrane. Well, but I prefer to always quote Dr. Irwin Mandel because I think this is the best way someone can have explained about saliva or defined saliva. That is, saliva lacks the drama of blood, the sincerity of sweat, and the emotional appeal of tears. So with that, let me just go forward. What exactly is this saliva diagnostics that we talk about? To make it very simple and straightforward to all of you, saliva diagnostic is nothing. It is just a study of our spit or saliva. And what exactly researchers do? Researchers get into this biofluid to search or look for certain elements and compounds that help them to diagnose and predict certain health conditions. It is not only about pathological conditions, but also about any physiological, normal physiological status. So that's a simple way I can describe what is saliva diagnostics. And let's see what kind of elements and compounds we can really find out from saliva. So I think we all know who are present here. It can be anything, it can be nucleic acids, that is RNA, DNA, whatever you can think. It can be hormones, it can be cells, it can be even immunoglobulins, it can be certain electrolytes, it can be even dissolved gases, and so on and so forth. So that's why saliva as a biological sample is it's a tool, it's a weapon, and I think the past 2 decades, much has happened in the global perspective. But it's time that we should take things forward and see it as a very very potential biological sample. Okay, and as you know that like the multifunctionality of saliva again makes it so more versatile because of the presence of these so many types of elements and compounds and whatever name them. And not only that, apart from the functions that we all know about digestive function, then it helps in mineralization and so on and so forth. But the presence of this antifungal, antibacterial, or rather I can tell that the antimicrobial components, be it immunoglobulins, be it any other inflammatory molecules, or which gives us information about the status of the body, the main status of the body. And altogether, that's why saliva has been very correctly called as the defender of the oral cavity. So now see, I'll give you a drone view of saliva. Why I tell this drone view, because you know what is drone. I mean to say that it's a drone camera, so we just see the overview. So whatever I have written here is just a piece. I don't know, you can add many things, but I just tried to give you a little information on what on areas saliva has been used and is being used, and in the future also there is very promising areas of research and development. So obviously, when it comes to identification of any particular gene or any regulatory aspects of particular DNA and all those stuff, then there is genetic and epigenetic research. Much work is being going on and has been done. Liquid biopsy is one thing where saliva is a standalone. It's like a boss there. And I'll tell you a little bit about all these things, and I'll just make go through the flow once I present more slides later on. Yes, human development. It's like if you talk about human development, then immense biomarker research has happened, not only for as such development, but the habit development, like feeding habits. With saliva, people have come up with certain researches with tests with saliva where you can actually identify the food habit, or the way a baby or an infant is fed, because there are many complications when there are premature babies. So that is one aspect. And also, when I tell human development, definitely another aspect related to our field, very much related to our field, is craniofacial development, craniofacial genetics. It's an immense area where you can plunge into. And yes, definitely, and work has started a long way back. And neurodegenerative conditions, like the commonest one that you can name, like Alzheimer's, dementia. So that area also much work is been happening, and now more focus has been put on that area, because I think Alzheimer's or dementia or depression, all these things are very kind of very common, becoming very common in the population worldwide. And coming to our fraternity, like if I talk about dentistry and with saliva, yes, many of you might be knowing that uh, periodontal medicine, in the field of periodontal medicine, much work has been done with saliva. So that is another aspect. But there are many other aspects where we can work with saliva, and this is not the end, or this is not the nutshell. I mean, this is not the picture what I'm showing. It's more than this. That is what I meant to say. And another very
मुझे आवाज समझ आ रही है ना ठीक है
Okay. Okay, so another very important area is stress biology. So there also saliva has immense applications, and many of you might be knowing about the cortisol, right, hormone. So salivary cortisol is always, all-time favorite. And drugs of abuse, identification of drugs of abuse, or estimating addiction, then differentiate. And I'll tell you a little bit of interesting stuff also little later on, that you can also identify between, exactly pinpointedly identify between a smoker and non-smoker. Maybe you will be knowing about this, but then still I thought it will be interesting to just tell you all these things. So that versatile is saliva. With this slide, I wanted to show that what not we can do. I mean, what we are doing, not that, but what not we can do. We can do everything with saliva in that way. Well, bringing you now to this is a very vast field, okay, but I just tried to sum up it in one slide. The world of omics, like you might have, recently you have been hearing, or those who are into the field, they might have known about genomics, transcriptomics, all the omics. So I have put all together. So what is this? This is omics is nothing but is just a study of different saliva biomarkers. Now, already we know that what are the elements or compounds, or what could be the possible biomarkers. It can be proteins, it can be DNA, it can be microbes present in the microbes present in our saliva. It can be anything. So in that way, there are many omics which has come up at present, but these are the parent omics that I can just tell you. So
Dr. Gargi, sorry to interrupt you. Can you increase a little of volume of yours? There are people who are complaining about it. Little more. Yeah, please continue. Sorry.
Should I increase the volume of my computer?
No, you can keep you can be a little close to your mic.
Yeah. Okay. Okay. Just give me 1 second. Let me just try to, because I have just reduced the volume. Just give me 1 second.
You were audible. It's not that you were not audible, but people are there. For me, it was quite fine, but people are there who are who might require a little louder. Yeah, please go ahead.
Is it fine now?
I think you are very soft spoken.
Okay.
That comes as a disadvantage, yeah. Huh?
Okay. Well, so I hope it is audible better now. Okay, so I was talking about omics, that is saliva biomarkers, how we demarcate them into this parent classification, I can tell, not classification, I mean, you can just group them. So what is genome and epigenome? So when you want to study about salivary DNA, or maybe any gene, or a group of genes, or you want to try to identify the genes or certain regulatory factors related to the DNA, or like, for example, transcription factors, or any genomic biomarkers, anything related to DNA. It can be a gene, it can be a non-coding part. So that comes under genomics or epigenomics. All right. Then transcriptome. So transcriptome is nothing but it's the study of the entire population of mRNA, of the RNAs, that is ribonucleic acid. So RNAs can be again different type, as you as you might know, but I'll just add on here, like, for example, you know about mRNA, you might have heard about transfer RNA or tRNA. Also, you might have recently heard about many things going on with regulatory RNAs, like, for example, microRNAs, silencing RNAs, siRNAs, miRNAs. So all those stuff, if you want to study, then that's transcriptomics. Proteomics is obviously when I'm trying to study the protein component, so it can be immunoglobulins, it can be enzymes, so anything that is protein that I'm trying to identify, or I'm trying to do some study or whatever, I'm trying to study those proteins. So that is proteomics. Metabolomics is also a very interesting area. Metabolomics is something is related to the metabolites. Like, how do I explain? I'll give you one example, which I think everybody of us understands. It's like, whenever we eat something, so it gets digested. So the glucose is the most simplest form of the complex carbohydrate. So if it is, so anything related to a complex, like suppose you are chewing starch, okay, and then what will happen? Ultimately, you get glucose. That's a monomer, right? So that's a metabolome. So for every different things that we eat, or the food that we are having, so how the cell metabolizes, basically metabolome or metabolic element, that's the last part that you get after a process is happening inside the body. And if you want to study that, then that is metabolome. It's a very fascinating field. It's a totally not different, but you can actually, it's a very vast field where you can actually study the metabolomics, and you can predict systemic diseases. You can predict just by seeing the profile. You can say how is the lifestyle of that particular person. So that's a very now upcoming area. And yeah, microbiome, definitely it's the study of different types of, we all know that the entire human body, including inside the mouth or in the saliva, even on the skin, inside our gut microbiome, everything we harbor many, so we keep that symbiotic relationship with different microorganisms. And if you want to study them, particularly if I talk about saliva, then that's microbiome. It's pretty simple. So let me go to the prominent technologies for saliva analysis. Just excuse me. Okay, so see, there are many things, many techniques, many things, but what I try to do in this particular slide is that I'm just trying to give you an overview. Like, what technologies can you use when you're thinking saliva as a biological sample or sample for test? So it can be immunological methods. I think many of us know about the very versatile and very commonly used technique, immunological technique, is ELISA, that is enzyme-linked immunosorbent assay. You might have heard about immunofluorescence. You might have heard about flow cytometric assays. And like, for example, there are certain methods where you can actually understand if you have a mixture of samples. Like, for example, if you don't know, if it's an unknown sample to you, it's a saliva sample and you don't know what it is, and you want to understand how many, what are the different types of maybe proteins are there, or anything. So then in those cases, when you really look into separation methods, you can focus on something called as mass spectroscopy. Okay, and then there are different types of mass spectroscopy. These are high-end technologies. Very simple technologies. When we come down to electroanalytical methods, like our very own colorimetric analysis, spectrophotometric analysis, they are very simple but very robust technologies. I'll shortly give you an example, and I'll try to justify what I'm telling right now. And apart from that, the molecular technologies that we have, the most common ones that we are using. Also, there are many other, but I have just pointed out some of the most prominent ones that are, as you know, PCR, that is polymerase chain reaction. Then there is next generation sequencing, microarray, all this stuff are there. So I don't think this is, it will, I mean, if I start explaining each of them, it will be very lengthy. So it is just that you get a snapshot of everything. I mean, I'll be able to do that particularly in this session. So now you know, you have all the things, right? Two things you have, right? Like what you want to analyze. So you have a list. You have proteins, either RNA, DNA. So you have all this stuff. So now you know about technology. So you have the tools. Now what you want for anything, for any sample to be analyzed, we need three things. First is the sample collection. We have to collect the sample, otherwise we don't start anything. Second is we need to understand the concept of the technology. What do I mean by this? I mean that suppose today I'm trying to analyze salivary DNA and I want to do it by, say, PCR. So if I'm doing by PCR, then at least if I don't know how to do PCR step by step, at least I should know what's happening with PCR. What is PCR? What's the basic concept? That is what I meant by concept of the technology, because then it gives you a broader vision and you can plan accordingly. You can plan your study, you can analyze your study. And the third thing definitely is whatever results we are getting, we have to interpret that result. So here I want to add one thing which I think I tell everywhere, because that is my perception, my own perception. That is, see, sometimes I have heard many people saying, okay, I have done this study and my results are negative. It is not positive. See, in research, there is nothing called as negative or positive. Every result is a result. The way you are interpreting that, I mean, it depends on how you're interpreting it. Okay, so I don't think there is anything positive or negative here. Okay, because we really don't know what is going to happen. We are trying to search that only. So even if you are copying somebody's protocol, you are changing something and doing, so that change you don't know, and in the downstream might add on to many other changes that reflects in your results, and you are expecting something and getting into that particular work. Please don't do that, because if you're expecting, then the question of results being negative or positive comes, because that is how I tell. Okay, so coming back, we have three things to know. That is saliva sample collection, because today we are talking about saliva, and concept of the technology, and results interpretation. So I'll just try to explain you with giving you some examples. But now we have, technology has advanced a lot. So I'll just show you, okay, in this perspective, I want to just mention that I'm coming up with the 2 months online certificate course from 14th of August, particularly based on saliva technologies. Maybe I'll be sharing more details later on. So I just wanted to tell this in this perspective. Now coming to saliva sample collection. Okay, the most common method of saliva sample collection is passive, we all know, and mainly most of the cases that we do is whole saliva. So these are different advanced tools that are available now commercially, which you can use for different purposes. These are all for whole saliva collection. Like for example, if you see this particular one where my cursor is moving, this is used for when you want to analyze salivary DNA maybe, and you need a larger volume of sample, like more than 1 ml or 1.5 ml, like that. And this is particularly one with which you can actually collect samples from very small babies, that is infants and children. And something like this, which is a bigger one, you can use this for teenagers and adults. And another fascinating thing I'll tell you, because sometimes you might want to not waste your sample at all, because sometimes the type of work you do, or the type of sample, the people or the subjects you select, from which even getting them again and again for sample collection might be difficult. So your intention might be that from one single sample, you want to analyze, or you want to collect both proteins and RNA components. So there is a very interesting device here, which these are all saliva collection tools. Okay, so what it does, it has a filter bifurcation unit. So at the point of saliva collection, that means right at the number one step, what you are doing by using these tools are, you are just subtracting the possible areas of errors that might creep into your results, because right from the sample collection starts your journey, because your results will reflect everything, whatever errors, whatever bias are creeping in between. There will be manual errors, there will be many other things, but this helps a lot. So so this bifurcation unit, it removes cells obviously, and what it does, when you after collecting, when you just press it's kind of a syringe like thing. Okay, and this is the cotton pad, it's absorbent pad, not cotton, but it looks like cotton. And if you just press this and when you collect in the pen drop, it will automatically bifurcate into two, and in one you get the total RNA, and in another you get the total protein. So that wonderful is the technology now. And suppose you are trying to work with you are specifically looking for, salivary DNA, and maybe you have to transport it to somewhere else, and you really need a robust method of transporting it also. And well, with DNA we don't have any problem, because DNA as a molecule is very, very stable, uh, and it will not degrade as such. So with this kind of a device, you can actually collect. If you see this, there's a vial. You can collect saliva as well as this kind of tooth structure that you are seeing, this kind of a handle. It also allows you to take scrapings of the buccal mucosa. So you have the entire picture inside that vial, and you're ready to go to ship it if it is required. And if you see, I have put original. This is a photograph of DNA that has been analyzed, that has been collected after collecting the saliva sample with using this particular device. And then the DNA has been extracted from the saliva using a particular saliva collection, DNA collection kit. And then there's a technique called as agarose gel electrophoresis, which we do for analyzing DNA or visualizing DNA, RNA. We do that. So in that, then a photograph is taken. What I just wanted to show you here, if you see, is a very purified form of DNA. So just imagine at the very point of collection, just after collection, you are doing extraction of the DNA. So you get a very, very pure sample. Your life becomes easy when you take this pure sample to your further whatever analysis or techniques you are using. So that is importance of perfect, or not perfect, but correct, using correct tools for saliva sample collection. Coming to the concept of technology. So here, before speaking into molecular technologies and all, I would like to highlight about the electrochemical analytical techniques, particularly colorimetry and spectrophotometry. So see, this is how a colorimetry looks, a very, very simple one. And I hope many of you have been seeing this. You have seen this in your institutions, or maybe in some biochemical labs, some big box kind of thing. But now these have become very sophisticated. It has come up, but initially it looked like a big box. Okay, so basically these two, colorimetry and spectrophotometry, are kind of it works in the same principle. Basically, it works in the principle of diffraction of light, and I'm not going to all those details. The main principle is that the main difference between these two is that spectrophotometry is kind of little higher end compared to colorimetry, and colorimetry works only in the visible region of the electromagnetic wave. I mean, how to tell you that? I mean, you know what is VIBGYOR or infrared UV, right? So that is what I mean. So it's only works in the VIBGYOR, because these are certain things. Because when I'm talking about light, so you understand it's all about wavelength. So basically, it gives you qualitative as well as quantitative estimation. So you get some reading like this. You set a particular wavelength, and you try to, that is what kind of light is going to come in, and then you check your sample. And when you see in this photograph, this is how the inside, actually this is the inside picture of the machine. And there are two types of spectrophotometry. Again, this is nothing. This is single beam, and double beam means very simple. In single beam, you can do one sample at a time. Like, for example, you have a control and a test sample. So first you have to put the control, then take the reading, again take it out, then put the test sample, and then take the reading. So that is how you can do with single beam. Double beam is that you can actually use two samples together. So in once there will be two slots where you can put that kind of a tube with your sample with a control in one tube. So it remains like that, and then one by one, how many ever test samples you have, you can just put and take the reading, and you can do the calculation. So that's the difference. It's very, very simple technique. There is nothing, even technologically also, there is nothing anybody on earth can do this. Okay, and now I will tell you how robust is this. Let's take an example. Let's say that we have a test scenario where we are trying to determine the psychological stress level in final year BDS student. Okay, I mean, everybody will agree that in final year, all of us who have gone through the journey of, or the grinding of BDS, we know what's final year. So let's see if this is the test scenario. So if I'm trying to, I'm trying to make you understand, to relate what I was telling previously. All right. So what would be my most easily available biomarker? What is most abundant? Saliva is known for its digestion of carbohydrates, right? So salivary amylase, alpha amylase, is one of the most available thing that is there in the saliva. So let's see the salivary. I will just try to analyze salivary alpha amylase. And definitely literature has this that there is a direct correlation where salivary amylase increases when there is increase in stress. So all this literature search I do, and then I come up that I want to test salivary alpha amylase. So how do I do that? So what type of assay is this? I'm trying to estimate an enzyme that is a salivary alpha amylase. So this kind of technologies or techniques like electroanalytical techniques, like I was telling about spectrophotometry, colorimetry, these are very good for enzyme assays. What will be the substrate? So as you know, that amylase will break starch right into smaller molecules and ultimately to glucose. So our substrate will be starch iodine. So what do I mean by that? So what would be the basic principle? So the basic principle of this particular technique is that if either the substrate or one of the reaction products that can be colored, because that is what we are trying to measure. So we can measure the intensity of color. I mean to say whether the color intensity is increasing, or we can measure even the disappearance of the color. So what's a very simple principle is that, suppose when I add iodine, okay, with starch, we all have done this experiment, I think in biology of class 12. You get a deep blue color, right? So that's starch iodine complex. Now, if I am adding enzyme amylase, starch hydrolysis will start, right? And then the blue color goes off slowly. Um, so that is my basic principles. Very simple. Now, if you see, I have drawn a graph here. So what is this? In this kind of estimations, what you have to do is, generally it is done like this. It's a very simple thing. Here, first of all, I'll take human amylase. It is available. And then I estimate at different concentrations. If you see, it is in the x-axis, it is concentration. And this is OD, means it's a particular wavelength in which I'm getting the readings. And this y-axis is giving me the readings. So these dots are nothing but readings. Like, for example, if I take, okay, let me just take 1. So if this is 1, then it comes near about, the concentration is like this. So if you just see, just by joining these two dots, I can understand what is the concentration. So what I'll do, I'll take a series of known concentrations from lower to higher, and I will take all the ODs and I will plot a graph. So that becomes my standard reference. Now you tell me that with any sample what you have, okay, like you have taken student sample, you don't know what is there, you want to see what is the concentration of salivary amylase, because that will give a picture of what kind of stress level is low, is it low, is it normal, or it is very high stress level for that particular student. So you just have to take your test sample, put it, get the reading, take the reading and put it in the y-axis, come down and see where it is going. So you get the concentration. So this is that easy. And well, there are some calculation that is not required to be discussed in this. I'm just trying to give you the simplicity. And now one more important thing I want to tell you. For anything we need money. We have to think about the budget. I'll tell you that with what budget you can do this kind of study. Okay, so here is the approximate budget for this project. If you're doing 10 saliva samples, okay, 10 is a good number for a pilot study. So if you are buying a colorimeter, you can do it with a colorimeter. Spectrophotometer will cost you a little more, but I'll tell you. So if you are buying the colorimeter for the first time, it will be, the lowest one is around 15,000, and obviously there will be differences. You can go high up in the scale, but I've put the most, the nominal one. You need 10 saliva collection devices, so that will cost you around, say, 6,000. There are approximate prices. Just for example, I've given. Then the iodine solution, there's a chemical grade iodine solution. Don't buy povidone iodine, that will not work. It is glucose iodine. So that is like a 30 ml bottle will cost you around rupees 2,000. But from that, you need only 3 to 4 drops, I think, 4 drops for one sample. And maybe some other miscellaneous, your consumables, this, that, okay, that will be 2,000. So your total costs come to 25,000. All right. But if you just tell me just to consider one thing, if you have this already, or the spectrophotometer is lying in your institution, I'm telling particularly for academicians here. So that means you don't have any instrument, you're not investing in the instrument. So your actually your even your cost comes down to rupees 10,000 or lesser, because here I have taken the prices in a little higher end. So you can do a research, you can do a wonderful study. It's a very competent study that you are doing here by understanding the psychological stress, because if you think the implications of this test, you can actually know what stage that person is. Maybe apparently the student smiles, does everything, but if really the stress level is high, you might consider to, give at least certain guidelines or some counseling or talk to that person. So it makes a whole lot of difference. I'm just trying to give example, nothing else. So now concept of technology, okay, let's see what's PCR. As you know that in PCR we have, it's a molecular technology, very common one. So if you see in this GIF, what I've put in this animation, there are 3 steps. One is denaturation. First, in this step, it is a very high temperature. You have all the raw materials for the PCR. That is, you have the ATGCs together, you have some enzymes, okay. So first the DNA has to, it's a double-stranded molecule, so it has to go apart. Then something called as primers, which are designed to according to the, which will match accordingly to the part of the DNA that you want to amplify. So that is, maybe this green portion is your DNA of interest. And then comes a step called annealing, where in the annealing step, the primers goes and bind to your target DNA. If you see, it will continue. Yes, this is the annealing step you see right now. So then extension, then this enzyme will help to extend. Okay, so that's PCR in very small way, I can tell you. And then another robust technology is next generation sequencing. Now there are many things has happened, but I'm telling you how it happens. Basically, if you have a target DNA, first we cut that DNA into small pieces, and then we give loads of nucleotides. I'm telling it in a very oversimplified and in a almost like a layman way, so that I can deliver the concept, because that is important, rather than knowing what integrate technologies are happening. So if you just minutely observe this particular GIF here in this animation, if you see that wherever there is a T will come. So when there is a correct pairing, one something flows, some light happens. So that is how you get a signal. For each nucleotide that is added, you get a signal, and you get one by one how is the arrangement. So this is where sequencing is a robust, because you don't know what is there inside the DNA. I mean to say how the ATGC is arranged. So what, I hope you know what is ATGC. That's the building block of DNA, that is adenine, thymine, guanine, cytosine. So that is what DNA is made up of. So when you take a unknown DNA, it's a very robust method, and that's why we can find out new genes, we can do many other things, and many applications. So I just wanted to give you a snapshot. Well, let's move on. And if you want to know more about the applications of sequencing into dentistry and what kind of wonderful studies you can do, then you have to opt for my another course that I'm starting on 18th September. That is also an online 2-month certificate course on dental genomics. You will get a wonderful insight. We'll also train you how to develop a project. So you'll be actually doing a mini project inside the same course. Okay, so let's move on. Now coming to the area of forensics, yes, here basically in forensics, saliva is used particularly for, I mean, more of, rather than a sample, we need to identify, right? So it can be two types of tests that we can do, where when we are trying to use saliva as a forensic tool, it can be presumptive test. That means we can establish the possibility that a specific body tissue or fluid is present or not. I mean to say whether saliva is present or not. And if there is a mixture of samples, and we really don't know what's happening. It may be dried or it can be many things, right? So we don't get that liquid saliva when it comes to a specimen. When saliva is a specimen, that's the correct word I must use. And yes, confirmatory test, whether I can confirm an identity, or I can compare and tell that if it is a paternity test, or whether that's the, it's a relation between the criminal and the victim, who is the criminal. So all these things. So there are certain interesting tests which are available, but unfortunately, I'm not aware that they are available in India. They're not, as far as I know, it's not available in India. But if anybody of you have any information, please, you can always share here. So again, that's something called as FADEVA's forensic saliva test. Again, it's a very simple one where I'll show you a small video. Here, the basic principle is again that they check the alpha amylase activity, and that stains. If it stains, they use something to identify whether alpha amylase is present or not. If it stains, that means presence of saliva is there. And then, even if it is dried, you can scrape it, and you can actually you can take out, and whatever you want, you can take out. For particularly, you can take out DNA, because that is one most important thing. That salivary DNA is one most important thing, or maybe from the blood when you take out DNA. DNA is one most important tool or molecule that has been analyzed from years in perspective of forensics, as far as I know. As I mentioned that I'm not an expert in this. Okay, let's see this small video. This will show you what is Fadeva's test. Okay. So, okay, so that was Fadiba's test, and I thought this will be something useful. So there is something called as rapid stain identification for saliva. Is the same principle is same, that is again alpha amylase here, but this is a strip test, just like you know about pregnancy test. So it's just like a strip test, and you can get the results in 10 minutes. And the best part is, if there is any animal saliva also present there, then this particular test will help to identify only human saliva. And there is, if along with any other samples like blood or semen or anything, any other biological fluid, if it is present, then there is no cross reaction, because it will not wrongly identify the amylase in the other sample. It will be only specific for saliva. And the detection limit, that means that as less as 1 microliter of, if you have the sample, it will help you to detect. So that is another tool. And well, this I found very interesting here. This is a test with this is based on the concept that salivary glands, there is thiocyanate electrolyte or an ion which is present. So in salivary glands, the ions appears to concentrate more. It's much published in the literature. You can go back and see. And there was a test called smoke screen, and this is something which is with which you can. This is a advanced version of smoke screen, where you can actually identify whether a person is smoker or non-smoker, or even the extent to which one person smokes. Well, if you right away go and search in Amazon, you will see this test available, but it's written that is only available in USA. My point is, can we not develop like this in India? So please think about it. Okay. So now coming to the genetic technologies, where in forensics, I hope many of you might be knowing about DNA fingerprinting and profiling. Well, DNA fingerprint is generally, it's done from DNA, and it's done from blood sample. First, the DNA is cut with certain specific enzymes. Then we do electrophoresis to separate the DNA fragments according to their weight and length. And then we transfer them to a membrane, and we incubate with some label probes, and then we take an X-ray photograph and we try to analyze. So that is one thing, but there are certain disadvantages. Now we have moved a little ahead with molecular technologies being applied into forensics. We do DNA profiling. So what is that? Here it can be saliva, it can be blood, and what we do not cut the DNA. We take it and we label them with the primers, and we do PCR, and then we electrophorese, and then we visualize, and it comes as peaks for each of the labeled STRs. So, well, let me tell what is STR. STR means it's a tandem repeats. In the whole human genome, there will be very, very small repeats, which is repeats means like these are very small, 2 to 4 nucleotides. Okay, like TA, like that. It will be repeated in certain intervals. So that are called as mini satellites. So we identify mini satellites with this. If I tell you the results, so this is how we come to the point where what I was telling, that we have to do result interpretation. So if you see this, You have got a sample here and you did DNA fingerprinting, and there were two suspects. So if you see, it entirely matches to the crime scene thing and the suspect. So you can go ahead and tell, okay, this is the one, this is a criminal. And in DNA profiling, what you do, DNA profiling is more specific. And one more important thing in DNA profiling or STA profiling, the best part is that even if your sample that you're getting is degraded, because in a crime scene, there will not be, we don't expect that integrity of the sample, right? So even if it's a degraded DNA, it works. We can do the profiling. As you see here, the interpretation that maybe it's a parental test, or who you want to identify what it is, whether the mother or father, whether it's really the child of these two of the couple. So if you see, these are the STRs that has been identified, and definitely some of the STRs from the father or and the mother both will be there. At least 50% will be there in the children. So same way, you can see here that number 5 is from the mother and the 7 is from the father in the child. So that means this is the child of this, the child belongs to them. So this is how we do DNA profiling in forensics. Well, another interesting thing I'll tell you in forensics, that is, you can do sex determination or gender determination. It's a very simple technology. I hope we all know what is amylogenin. It's an enamel matrix protein, and what you can do in what happens in X chromosome and Y chromosome, because that is how the male and female is segregated, right? It is 2 X's for females and X and Y for males. And then if you just take saliva samples, okay, and in males, if you just analyze the amylogenin of a particular area of amylogenin, okay, I'm not going into that details, but if you analyze, then in case of females, you just get 1 band. In case of males, you get 2 bands. That's simple it is. Okay, so these differences of amylogenin gene expression straight away gives you an idea of gender differentiation. So that is how you can do. Okay, coming to almost to the main area, or maybe I'll try to tell you the potential areas of research with saliva and dentistry. Well, I have pulled up all these because these are very good areas to work on. And if you see the global perspective, you will find hundreds, and the literature is filled up. But I think if we can do something in the population of India, and it is happening, it's not that it is not happening, but it will be really wonderful to address these areas, particularly dental caries susceptibility. We do something called as genome-wide association studies. I will tell you all these things with one example. Craniofacial genetics. Third is oral microbiome, where you actually, the oral systemic link, like we all know that there is a correlation between the oral health and the systemic health. So that is one area. Third is definitely oral cancer, but early detection. That is one of my most, I'm a diehard fan of this particular area, because I think for any cancer that matter, early detection is the key. Then whatever we do it. Okay, so there comes the role of liquid biopsy, exosomes and all those stuff. So let's see. Let me just give you an example of some studies that have been done, so you will understand how important it is and how this can be done. So what is genome-wide association study? So one of the studies, they found that they found actually 47 risk genes. Which 47 areas in the genome where the genes were responsible for the susceptibility to dental caries. Okay, and how they did this is that there are certain there's something called as gene lifestyle interactions in dental endpoint, the GLIDE. This is a kind of a consortium. Like you can go, it's like available to everyone. You can go and you can search your if you have some sequence with you, or if you want to search whether your gene is how it is happening, or you want to know what is there inside your sample. Then you take all the information, you take the clinical symptoms, you analyze DMFT, DMFS, all those things you can do. And this is how they did. And also you have to take the genetic information. I'm telling it in a very crude manner. I know you might not get the entire thing, but what I mean to say is that genome-wide association study in one line means you search the entire genome for a particular disease to find out that what are the different genes present that might be most important for this particular disease. That is what is genome association study. So dental caries susceptibility is one area which is a very good area to plunge in. Second is craniofacial genetics. I will tell you a very interesting study that has been done here. Why I'm giving you these examples, it will be easy for me to tell the concept. So one of the studies in craniofacial genetics, in one of the studies, a very interesting thing they did is they analyzed certain microRNAs, and they found specifically 3 microRNAs. I have mentioned it in the slide, like for example, microRNA 141, 223, and 324 maybe with 3P something. Okay, so what they found, these specifically 3 microRNAs are dysregulated or deregulated between the study group and the control group. So what was their study group? Study group was cleft lip and palate patients. Okay, so they tried to record the differences between the expression profile of the microRNAs between these 2 groups, and then they also saw that through the literature that particularly these 3 microRNAs are again involved in the, or they regulate the expression of certain genes which are responsive, which are very much correlated to cleft palate and cleft lip development. And so that means the differences in regulation of these particular microRNAs between the 2 groups of salivary samples suggested that these molecules can be prognostic biomarkers. That's the key of this study. So if you are thinking that this is something which is very complex, it's not at all. Now technologies are so much available, not in US, not in Western world, but in India. Everything is available. It's just that you need to just focus on and take up these areas and come up with wonderful, information and technology, so that would be benefiting to the society. And oral microbiome, we all know about how the periopathogens, like for example, many of the studies have shown, and particularly one study have shown where 4 periopathogens have been independently involving in lowering the serum levels of high density lipoproteins, and that is why the same thing has been responsible for increased risk of atherosclerosis. I think this is a very known fact to many of us. And as I was telling, oral cancer early detection, yes, you can think of, definitely comes here, liquid biopsy. So you can think of analysis of microRNAs, exosomes is wonderful area which you can plunge in. Yes, but you will need a little bit of funding and budget for this kind of programs or projects, because these are, you need to involve high tech or high throughput technologies, obviously. Circulating tumor DNA, that is ctDNA. So all these things are very, very good areas to think on, to build up or take up more research. Well, with this, I have one more thing to share. I'm very happy to share that I could successfully start International Summit of Saliva Symposium India, or SALSI, in the year 2019. And this year I did, just before the pandemic, I did it in IISc Bangalore. So this platform is something where all world leaders come together who are working in different areas of saliva. It's a very, very versatile and interdisciplinary platform. It's not any it is not a conference, or it is not a meeting where we focus only on one subject, but people from different avenues. But one common point is working with saliva comes in, and I hope I'll be able to do the next one also. But let's see how the things goes this time. And in this summit, I started a program called as Kriya, dedicated to my dad. So that's Kamalesh Roy Research Idea Award, to promote undergraduate research in India, where many people, many students, they are all undergraduates. If I would have been able to show you those, those presentations, you would have wondered how robust ideas they have. And it is not only about idea. Some people came up with already, they are doing some projects. So I just, it was a very good, very good presentation, the way they presented, and we trained them before that how they should teach. They will have only 4 slides and stuff like that, just to give them a feel that when you really go and ask for money or for grant or fund, how is that done? So, and then at the end, we just, the winner, we selected one winner, and we gave some cash prize to them. So that is what I started, and I hope I can make it little big with some more support. And I hope this will really push the young generation into to think in a different way. And they are, I'm telling you, if we can harness their energy, they are fantastic. They are fantastic. You just can imagine what they have presented. So that's the website. You can go and visit whenever you're free. And well, I'll tell you a very interesting thing now. That is a pathway to entrepreneurship with saliva. In one word, it's bioentrepreneurship. So what is bioentrepreneurship? Basically, it's it is the bringing together two things. It is little different from entrepreneurship. Entrepreneurship, you just take, you have to take risk of the capital, you have to take the capital risk maybe, but here along with capital you also have to take the risk of technology. So entrepreneurship is the actually the flow of innovation from academia to industry. So why don't you all start thinking in that way, that whatever work you are doing, whatever small projects you are doing, that you can come up with your own product, and own tests that are really necessary to fill the gaps that we have at recent time in the clinical area. So how you can do it? You definitely need fundings, or you need collaborations and money. So these are the three ways that you can do. That is government grants or funds. Then there are contract research organizations, and then self-finance. Definitely, that is one option that we call as bootstrap. And for government grants and funds, there are many things. And I can just focus on one thing, that is a non-profit section that has been set up by Department of Biotechnology, that is called as BIRAC, Biotechnology Industry Research Assistant Council. So BIRAC has many types of grants that are available. Even if you are an academician, you are an individual researcher, you are a clinician, you just need to be undergraduate to apply for this fund, obviously with a very robust idea and a very you should be passionate about what you are doing. Only then you should jump into these things. Okay, so that is how you can do. And there is a whole process. It takes little time, you need patience, but that's nothing. I mean, that's the way, everything has a protocol, so you just have to follow the protocol. And with us, we help you in developing your, if you want to come into bioentrepreneurship, most welcome. We help you to come up with collaborative grants. If you have idea, if you have no idea, if you have done a small pilot study from any stage, we can take it up. We can help you with the product development, because we have different collaborations with different people, industry people, where it is necessary. Like for example, if you want to bring up a device, so you can take part of the biochemistry. The device people will take up of how to build up it as a device. So we as clinicians or researchers, we won't be able to manufacture a circuit. So that we all know. And then how you can commercialize them, how to follow up with your product, and how you can actually come up with your business strategy. So that is all in small things we do here. And other than that, from my way, I give research consultancies for academicians, particularly planning the project. That is very, very important, because if you can plan your project well, then the next steps just comes in the flow. And second thing is, as you know, that I am very much into this through the education and training. I'm trying to provide education and training in the field of dental genetics and saliva diagnostics, and also we have research services like any other laboratory. Okay, so with that, I'll come to the concluding part, and these are some of my voluntary scientific endeavors, and that's my website. You can see whenever you're free. It's www.geneden.com. And if you want, you can join our Academy Facebook group. You can just write the whole name, Indian Academy for Clinical and Dental Genetics, or you can just try to search with this hashtag in the Facebook. So here in this group, we do some things which, like we discuss some topics every day. I come up in live sessions every Wednesday. I try to maintain the pace. Sometimes I miss, and sometimes I bring some experts. Also, there is something called a Showcase Sundays. So in Showcase Sundays, any member of the group, if they want to come up and highlight their personal talents, or about their professional life, anything, they can come up and showcase. They can promote themselves. Apart from that, I think already Dr. Raman has mentioned about Eclectica. That is what he was telling. That's the interview show I do in my YouTube. So please subscribe my channel, and also you will find certain free webinars, particularly related to saliva in different areas. So that's Saliva Annex. Those are all there in my YouTube. So whenever you're free, please go and visit. And you can always connect me if you can mail me at dentalgeneticsacademy@gmail.com. And thank you so much. I think I ran a little ahead. I'm a little ahead of, I took too much of time. And thank you so much. And it was an honor speaking here today in front of this august audience. Okay, so thank you so much. I think it was, I was supposed to talk
Thank
about
you.
45
Thank you so much.
minutes.
Thank you so much. Thank you so much, Dr. Gargi.
Thank you.
We'll be moving on to the question answers, maybe 10 minutes. And
Yeah.
I think Dr. Priyanka will take over from here.
Sure.
Gallery view. Yeah.
Dr. Gargi,
Yeah.
loads of thanks for a
Thank you.
very wholesome presentation, I should say. You have covered almost every aspect that we could think of, right from diagnostics to whatever potential applications we have, even genomics, omics, everything.
Yeah.
So, in fact, you kind of ignited minds of everybody, and they have been pouring questions ever since. So, you have a lot of questions to answer now.
Okay. Okay.
So, really, really thanks a lot. And Dr. Abhishek Das, the first question he has asked about is about forensics. And he wanted to know that how can we collect and preserve saliva sample during autopsy, and also any research possibilities of salivary diagnostics and post-mortem examinations.
Okay, I'm not an
From
expert
the
in
background of
forensics.
forensic, yeah.
Okay, I'm not, I don't have any expertise in forensics, so I gladly admit that. But yes, I can definitely add on how you can, any saliva sample that you're taking that you can store. For particularly, it depends on what you are trying to analyze. If you're trying to analyze salivary DNA, for example, then DNA as such as a molecule is very robust. Like if you just collect it in a tube and keep it, apart from your microbial growth, that's going to happen. But DNA can be stable for 1 month if you just keep it. But it is better to keep it in a 4 degree. At least you give that. But if you are trying to analyze about salivary RNA or proteins, that's a big no. You just have to give the minus temperature immediately as possible. So that much I can tell. So I really can't help you with your exact answer because I don't have that expertise.
Because regarding storage, Dr. Deepika had also asked, is there a correct way for collecting saliva, storing saliva, any room temperature, any time that you have to, for how long you have to store it? Is there any criteria, any specific protocol regarding it? Or it
No.
depends on what molecule we are going to analyze
Yes.
in saliva?
Absolutely. There is no protocol, but that the best and safe thing is that once you collect the saliva, because if you are standing near your instrument and if you're immediately doing the test, that is fine. But if you're not doing that, go and first store it in the normal refrigerator, that is 4 degree. And for particularly proteins or RNA samples, like if if you are looking for analyzing salivary RNA or proteins, then just go and keep it in the -20. That's the deep freezer that we
Any
do.
room temperature that we have to take into consideration, or any time of the day that it has to be a morning
No,
sample or an
no. That depends on what analyte you are trying to
what
analyze.
analyte? Okay,
Yes.
okay. So it depends on
Because
the analyte.
because many analytes, many molecules or many enzymes, whatever you are trying to analyze, they might have certain diurnal differences. Like maybe that, say for example, calcium. You want to analyze salivary calcium, so calcium will be less in the night or more in the morning. So it all depends what you are trying to analyze. So your analyte will give you that discretion, and then you have to standardize your protocol yourself. That's the one lacking point in with saliva, that we don't have a gold standard to follow.
Standard protocol. Yes.
Yeah.
So related to that only, I think then you kind of propagating that point of care analysis would be the best ones. So are there any protocols regarding the point of care analysis? That's what Dr. Aman had asked. That how do we plan a point of care analysis for saliva?
Okay, you want to come up with some
Yes.
some test.
Some
Okay,
test.
so
Which is
yeah.
chair side, because we are dentists, so it is beneficial for us if we can do anything which is chair side. So we
Yes.
don't have to even store it. So that is also not will be a problem.
Well, that's with any, not only about that's with any analyte or anything with saliva. Meaning to say that, for example, say for example, I want to develop a device which will give me a qualitative estimation of whether a person is, whether the person is into towards the oral cancer or not. So again, it depends whether you want your test to be only qualitative, or you want to have a quantitative also, or you want to have both together. So that is one area that you have to think at the first go when you are trying to plan. Second thing, definitely you have to look into the when you're coming to develop for a point of care technology. Here one thing I should mention, that the grants or thing that the industrial grants that you get, mostly if you are going with a project which says that, okay, you are very passionate, you have a very fantastic idea, but you are trying to identify a new biomarker, you will never get a grant. Okay, because because the thing is, they will see the commercial feasibility of that particular thing. So the thing is, suppose what you can do, you can innovate. I mean to say, suppose 2-3 biomarkers has been well studied but related to a particular disease. what you can do, you can bring them together and bring up your own innovative idea. That suppose for example, you tell, okay, if I say that biomarker 1, 2, 3, 4, if this is the combination, then it really gives a prediction for oral cancer. Okay, I'm just giving you an arbitrary example. So that you made 1, 2, 3, 4 and not 2, 3, 4, 1. That is your innovation. Okay, so then for point of care technologies, basically, it is always good that I'm not telling that you don't go into finding new, but if you really want to come up fast with a solution, which is already the problem is existing, you have enough literature, then you just have to think in a different way. That is all. That much I can tell in this case. Yeah.
Dr. Varun had a question regarding where to buy the saliva diagnostic kit and how much is the cost of the kit.
Yeah, so the tools that I showed you is available with my company, because that's the thing that with a passion I came up with this company, particularly focused to saliva diagnostics, because these tools are from our parent company Oasis Diagnostic USA. So we are the we are the exclusive representative in India. So you can get, it is available with us. And secondly, the cost, it depends. It ranges from about somewhere around 500 to almost 1200 or 1300 per unit, depending on which you are selecting. And also if there is, like, it also, as you know, for anything, if it's a bulk order, so sometimes the prices comes down in that way. So yeah.
Okay, there's a very pertinent question by Dr. Aman and Dr. Anvesha regarding passive drool saliva. So is that the only, drooling, is that the only technique for a passive collection of saliva, or there's some other technique also?
No, that's passive drool. I mean, the way naturally the saliva is flowing. So
if we have generated some cough or something in the patient, that's not passive collection. That's something which is stimulated, right?
Yeah, I mean, I should not say stimulated. I mean, then you have more of contamination. It's not, you can consider, but then you will have nasal and bronchial secretions mixed up with your whole saliva. That
So
will be
what is the way to collect pure saliva? Is there any way to collect a pure saliva sample?
Yeah, pure means what is your analyte? What is your whether you want DNA or RNA or protein, as I was telling. The different saliva collection tools that I showed you. See, first thing what you have to get rid is we don't want mucin, we don't want cells. Okay, so now if you want exosomes, then exosome is a small vesicle, membrane bound vesicle, which actually exosome is something where during protein processing, okay, it will pull.
Ma'am, I will be very specific here. If we are going for an RT-PCR, what kind, and we want it exclusively on saliva, the question lies that it should be pure saliva. So how to go about?
Then you can use you can use any of these tools that will give you perfectly pure saliva. You will get what you want in that saliva, and all will be excluded what you don't want. If you want salivary DNA, then you have to use a specific collection device for salivary DNA. So that in that case, you don't want protein, RNA, cells, etc., etc. So all these things will be.
The only problem lies that when we collect it, the problem lies that it will be having phlegm, it will be having other respiratory secretions as well. So is there a way that only?
No, that doesn't matter. That doesn't matter.
I mean, it might not matter when we are going for a diagnosis, but if we are doing a study that it is present in saliva or not, purely in saliva or not.
No. It doesn't matter. It's not about the diagnosis. It's about when you're doing a study. So see, the thing is, you are collecting the molecule, the DNA. So whatever it is there, the first stage of when you take this sample, even if you mix soil and if you give me, I guarantee I can give you purified DNA because of the process of DNA extraction. Okay.
No.
Okay,
Actually, ma'am, here I think the question is that suppose you have a same molecule, you have that in phlegm also, you have that in saliva also, but you're doing a study where you're comparing both, or you just want
exactly
related to saliva. So
can
is
you
there any way to be sure that you are collecting just the saliva sample and not the phlegm sample?
No, if it is, see, if in saliva, when you want to take out saliva DNA, for example, okay, so then first step we do for any saliva sample, we need to centrifuge it. So whatever is unwanted, it will come down. In the solution will be the DNA, okay, and the proteins and RNA. Now, if you are not using any tools, I'm telling, if you are using any tool specific, then you will get a purified sample at the first go. That is what I'm telling. as a purified sample at the first go.
All right.
Could I answer? Could I answer your question?
Yeah.
Yeah.
We got it.
Okay.
I think we'll go forward. Dr. Weber has asked how to store the saliva nicotine kit. You had mentioned the nicotine. That's a news for me also, actually. It's a point of care for smokers. So I think
that's a strip test. Just like you get it as if you go and see in the Amazon, all the photos are given. It's just like a pregnancy test kit. You just can store it in the room temperature itself. It will be packed. It comes in some 5 packs or something, but that's available in US. So you have to develop so Dr. Baiva has to develop that so that we get it in India. Okay.
that would be great. Dr.
Yeah.
Nandini has asked a very pertinent question. Any difference between collection in children and adults? Saliva
Yes.
collection.
There is nothing much difference, but the thing is with adults will be more compliant as a subject, but with kids, with children, they will not be compliant. Okay, so as you saw that I showed one particular device. In case of saliva collection with those kind of devices, the length of the absorbent pad will be small in case of the children or very small infants, if I'm talking about. And also for adults, generally what we tell them to, like, if you want to really collect the pool of saliva, we tell them to keep their head forward, so that in the sublingually you get the saliva, and you then take it up, you put the absorbent pad and do it. But with children, you cannot keep them like that. They will fly off. And also with infants, they don't have their own control. So in case of children, we tell that tilt the head, tell them to tilt the head, and for children it will come faster. And then with that swab kind of thing, not swab, but that device kind of thing, if you put it, it will automatically soak for some time. If you keep it, then you can collect it. So that's a little small difference in technique. Yeah.
The last question, I think I'll just put it up.
I would like to add here that we go down in 3 minutes. So
just last question, which I don't think that Dr. Gargi can answer, because it's a huge question. What are the salivary application in COVID times? So I think you can keep it for your next lecture, because we are getting a lot of requests for one more lecture from your side. And people have written that what all they want to know about oral microbiome, and they want a detail. So they've not had enough of you, Dr. Gargi. So I think you'll have to be on this platform again. Thank you so much. And I think I'll hand over the mic to Dr. Deepika for a formal word of thanks.
Yes, Dr. Deepika.
Yeah, I take it as a great privilege to deliver the vote of thanks on behalf of my whole team, Innovative Pedagogical Learning. That's Dr. Aman, Dr. Priyanka, myself, Dr. Deepika, and Dr. Pooja, for such a stimulating talk on saliva diagnostics. I'd like to thank Dr. Gargi for covering such a vast topic, including all possible applications of saliva, and all in one composite lecture. Indeed, ma'am, this has opened up many avenues for us to work on, and it has introduced us to the world of bio-entrepreneurship in your terms. And like Dr. Priyanka said, the chat is filled with requests on specific areas that people want detailing on. They've just not had enough. So everybody wants more of saliva diagnostics.
Sure.
I'd like to thank I'd like to thank Dr. Goria also, who is the president of the Indo-Pacific Academy of Forensic Odontology, for allowing us to hold this lecture under the aegis of INPEFO. I'd like to thank Dr. Emilio for his partnership, with Association of Forensic Odontology for Human Rights, Italy. I'd like to thank Professor Jasuja and Dr. Neeraj Grover for their support of IACFI, that is the Indian Academy of Forensic Odontology. And I want to express my heartfelt gratitude to all the participants for today's lecture. Thank you all for joining us today, for being so active and